Today we study the topic meiosis. Meiosis is nothing but reduction division, right? Why reduction division has to occur? The living organisms in that the chromosome number, it has to be maintained in a uniform manner in each generation. To maintain the uniformity in chromosome number in each generation, meiosis division or reduction division, it is essential. Meiosis or reduction division, when we are saying what is reducing here, the number of the chromosomes, it will be reduced to half, and this meiosis always occur in a diploid cell. So meiosis occurs in diploid cell. Meiosis occurs in diploid cell, and this meiosis, it occurs in diploid cell to reduce the chromosome number to half, right? So 2n number of chromosome, if it is present in a cell, it will be reduced to n after meiotic division, right? So which all type of cells will undergo meiosis? Which all types of cells undergo meiosis? Which all type of cells will undergo meiosis? We will see. Meiosis, it occurs with diploid cells, we say. The meiosis, it occurs with zygote in thallophyta. In thallophyta, zygote undergo meiosis. So we can say zygotic meiosis occurs. Zygotic meiosis occurs, right? And we can see the spore mother cell, it undergo meiosis. Spore mother cell, it undergo meiosis, that we will say sporic meiosis. That is sporic meiosis. I'll give the example for this, okay? Sporic meiosis. Now, next one I'll say the gamete formation for that meiotic division, it occurs. That is gametic meiosis. Okay. So what is this? Zygotic meiosis. In the case of thallophyta, they are following haplontic life cycle we have studied, right? They have actually gametophytic plant body. So the gametophytic plant body or haploid plant body, they produces the gamete by mitotic division, that is haploid gamete. So the haploid organism, when they produces the haploid gamete, and haploid gamete, when it is fusing together, it will become as diploid zygote. That zygote, it will undergo meiosis to produce the spores, haploid spores. These haploid spores, actually it will undergo development in the form of a complete organism in thallophyta. This is seen in thallophyta. So I'm just writing it is seen in thallophyta. Okay. Now the next one, it is sporic meiosis. Sporic meiosis, for that the spore mother cell, it will undergo meiosis. You see, this zygotic meiosis, that is a characteristic feature of thallophyta. Like that sporic meiosis, that is seen in other than thallophyta, all the plants, they show sporic meiosis. So we will see how the sporic meiosis, it occurs. Bryophyte, in that when they are producing the gamete and undergo fusion, it will become a zygote, we say, and the zygote, it will develop a sporophyte. We have studied sporophyte, it will be having capsule. Capsule, it consists of spore mother cell, right? So I'm just writing spore mother cell for that, SMC. Okay, SMC is nothing but spore mother cell. Spore mother cell present inside the capsule will undergo meiosis. It will undergo meiosis and it become as haploid spores. Haploid spores. These haploid spores only will undergo development in the form of a haploid plant body. This is in bryophyte. Spore mother cell undergo meiosis and it produces the haploid spores. Now in pteridophyte, you see, sporangia consists of spore mother cell, undergo meiotic division, and it will become as haploid spores, and this haploid spores, it will develop as prothallus gametophytic plant body, right? So there also the spore mother cell undergo meiotic division. So spore mother cell undergo meiosis to produces the haploid spores. Haploid spores, now I am giving the example, okay? This haploid spores develop as haploid gametophytic plant body in bryophyte. What about the pteridophyte? You see, bryophytes. In pteridophyte, actually, the haploid spores which is produced will develop as the prothallus, that is gametophyte. That prothallus will develop the antheridia and archegonia. From there, real gamete will be formed by mitotic division, right? So here we can say the haploid spores produced in pteridophyte develop as haploid prothallus. Prothallus is nothing but gametophyte. I am not writing it again because we have studied the life cycle of this bryophyte, pteridophyte and all, right? So this is in pteridophyte. Now in gymnosperm, haploid spores produced from microsporangia. Haploid spores from microspore mother cell. Microspore mother cell. Microspore mother cell. I'm just writing mother cell for that. Microspore MC, mother cell. Okay, haploid spore from microspore mother cell will become as haploid. Sorry, haploid spores from microspore mother cell will form the microspores, or haploid spores which is produced from microspore, that is called as microspore, develop as pollen grain. Develop as pollen grain. You see, there also the microspore mother cell, it is undergoing meiotic division. Like that, megaspore mother cell also in gymnosperm. Okay, pollen grain in gymnosperm. Like that, the megaspore mother cell also we can say. And in the case of angiosperm, in angiosperm, I am not writing again microspore mother cell which is present inside the anther, that will undergo meiosis and it become as microspore. That microspore develop as pollen grain, and that is in gymnosperm and angiosperm, both the microspore mother cell undergo meiosis and it become as microspore undergo development as the pollen grain. Okay, so this is the way the sporic meiosis occurs. Sporic meiosis is a characteristic feature of plants. Now gametic meiosis. Gametic meiosis are seen in gametic meiosis. It is seen in animals, that is for the formation of gamete. Okay, so formation of gamete, it is for the formation of gamete. It is for the formation of gametes, and we can see this is seen in animals. Example, I'll just say in the case of human. Example, sperm is a male gamete produced from spermatogonial cells by meiotic division, right? So male gamete sperm produced by meiotic division in spermatogonia, or we can say sperm mother cell, spermatogonia or sperm mother cell. Like that, the female gamete ovum is produced by meiotic division in oogonia or ovum mother cell. Okay, so once again I'll say meiosis, that is occurring in the diploid cell. Actually meiosis is for what purpose? To reduce the chromosome number to half. Means meiosis always occur in diploid cells only, because in diploid cells only something is there to reduce, right? So the diploid cells after meiosis, we can say it is becoming as haploid cells. Okay, now which all type of cells undergo meiosis? Meiosis, it occurs with the zygote. Meiosis which occurs with the spore mother cell. Meiosis which occurs for the formation of gamete or gamete mother cell, in that meiosis occurs. So according to that, we can just see three different types of cells undergo meiosis, and that meiosis we have just classified as zygotic meiosis, sporic meiosis, gametic meiosis. Zygotic meiosis, zygote will undergo meiotic division for for the formation of haploid spores, and this haploid spore will develop as haploid plant body that is seen in thallophyta. Sporic meiosis, spore mother cell undergo meiosis to produces the haploid spore. Remaining all plants they show sporic meiosis. Okay, example if you are saying bryophyte. Bryophyte we know they are a haploid plant, gametophytic plant, and this gametophytic plant they produces the antheridia and archegonia as the reproductive structure. Antheridia produces the antherozoid, that is the male gamete haploid, and archegonia produces the egg which is female gamete, that is also haploid. How it is producing? As it is produced from the haploid gametophyte, it will be produced by mitotic division. So gametes are produced by mitotic division in bryophyte. This gamete, when it is fusing together, it will become as diploid zygote. That diploid zygote, it will undergo development in the form of a sporophyte. We have studied that sporophyte. It has three different parts we studied, that is food, seta and capsule. In the capsule there are spore mother cell. That spore mother cell undergo meiotic division to produce the haploid spore. This haploid spore, it will undergo development in the form of a haploid gametophytic plant body as the parental plant. You see, sporic meiosis only it has occurred. No spore mother cell present in capsule undergo meiotic division. Now, next one, pteridophyte. We have studied pteridophyte is a diploid plant body. Its reproductive structure is sporangia. That sporangia consists of spore mother cell. That spore mother cell undergo meiotic division and it produces the spores. What will happen to these spores? These spores will fall down, and when the spores are falling down, it will develop as a new plant body, that is from the haploid spore haploid plant body, gametophytic plant body, prothallus will be developed. That prothallus, it will form the antheridia archegonia structure that produces the gamete by mitotic division. We have studied these gametes, it will just fuse together to become a zygote. Zygote develop as a progeny. That progeny, it is a diploid progeny because parent is also a diploid plant, right? So there we can say they undergo sporic meiosis. Sporangia consists of spore mother cell undergo meiosis to produces the haploid gametophytic prothallus, right? Now the next one is gymnosperm and angiosperm. Gymnosperm, it has microsporangia as the male reproductive structure, megasporangia as the female reproductive structure. Microsporangia consists of microspore mother cell. Megasporangia has megaspore mother cell. Microspore mother cell undergo meiosis to produces the haploid microspore. This haploid microspore, it will develop as a pollen grain which is multicellular condition. Okay, whatever it may be, that microspore mother cell, it is undergoing meiotic division, that is also spore mother cell, sporic meiosis. And in the same way, in the angiosperm also it will occur. Microsporangia consist of microspore mother cell undergo meiotic division to produces the haploid microspore. From their pollen grain will be developed by mitotic division. So which cell undergo meiotic division? Microspore mother cell undergo meiotic division. Same way in the female reproductive structure of the gymnosperm and angiosperm also. Gymnosperm, the female reproductive structure is megasporangia. Angiosperm also it is megasporangia ovule, right? So there in gymnosperm, megasporangia, megaspore mother cell undergo meiosis to produces the megaspore. That megaspore, it undergo mitotic division to form the archegonial structure, that is its gametophyte or the female gametophyte archegonial structure. In that archegonia or egg, that is the female gamete. So there also we are seeing the megaspore mother cell, spore mother cell, sporic meiosis it occurs. In angiosperm again, the megasporangia consists of megaspore mother cell. That megaspore mother cell undergo meiotic division to produces the megaspore. That megaspore develop as embryo sac by mitotic division. So there we can see megaspore mother cell undergo meiotic division, not the gamete mother cell undergo meiotic division, right? So in the case of plants, the meiotic division, it occurs either with the zygote or with the spore mother cell. Now we can see gametic meiosis. Gametic meiosis is usually seen in the case of animals, where we can see the gamete mother cell which is present in the primary reproductive structure as testis or ovary, where we can see the sperm produced from the sperm mother cell. It undergo meiosis and it become a sperm. Sperm is a male gamete. So gamete formation for that meiosis, it occurs for with a gamete mother cell, gametic meiosis. So gametic meiosis, it occurs in the animals, and the male gamete, sperm produced from the sperm mother cell, female gamete, ovum produced from the ovum mother cell or oogonial cell. Okay, so this is all about which all type of cell undergo meiotic division. Now we have to study how meiosis is occurring in the living organisms. Okay, now we will see how meiotic division occurs. Meiosis, actually it is a reduction division. I say meiosis, it form, meiosis, it form 4 cells from the meiosis. Which cell? Means meiocyte, means the cell which is undergoing meiotic division. Okay, so one meiocyte, it undergo, meiocyte is nothing but the cell which is undergoing meiosis. After meiosis, it will form 4 cells. Okay, so here the meiocyte, it is always having 2n number of chromosome, and these 4 cells, it will be having n number of chromosome. Means diploid cell, it will become as haploid 4 cells, right? So this meiosis, it occurs in 2 stages, meiosis 1 and meiosis 2. Okay, before that we can see this will pass through the cell will, first step, the cell will enter into into interphase. So in interphase, the cell, they will be in preparation for the division. So there in interphase, they will undergo replication for their DNA. The cell, it will grow, and it will just increase the cell organelles, everything. So in interphase, it will pass through G1 S G2 phase, and it will become like the cell's chromosome number and content, it will become like 2n with 4C content of DNA will be present in it after the interphase, right? This we know already in mitosis, we have studied. Now this meiosis, it occurs in 2 stages, I said. So meiosis 1, actually it is a reduction stage. Meiosis 1 is reduction stage. In reduction stage, actually what is reducing? We said the number of chromosome will be reduced. So 2n, it will be reduced to n. So haploid number with 4C content of DNA will become as 2C content of DNA, because 2n number of chromosome, if it has 4C content of DNA, how this 2n number of chromosome is with the 4C content of DNA? Because each of the chromatin thread or chromosome, it is with its sister chromatid, right? So in the meiosis first division or meiosis 1 reduction stage, in which we can see the 2 cells will be formed. 2 cells, the resultant cells, it will be 2 cells. 2 cells will be formed with n number of chromosome, haploid number of chromosome, and 2C content of DNA. 2C content of DNA. You see, we have studied already, like there is no need of haploid number of chromosome. If it is present, there is no need of 2C content of DNA, only 1C is enough. And diploid number of chromosome, that need 2C content of DNA, like that we have studied. So here haploid number of chromosome with the 2C content of DNA, it is excess of DNA present in it. So the content of DNA has to be reduced, right? So meiosis 2, in which we can see it will separate the sister chromatid to reduce the content of DNA. That is just like a equational division. Okay, when I'm explaining that, you will understand that. Now you just understand it is a equational division, where we can see just like mitosis, it will be separating the two sister chromatids. In mitosis, actually, we have seen the centromere region from where two sister chromatids are getting separated into each pole. No? The same way, the equational division in which the chromosomes, it will be coming and arranging in the center region, and it will be separated into each pole. Okay, so again, 4 cells will be formed from these 2 cells. So 4 cells, it will be formed with n number of chromosomes and C content of DNA. You see, the content of DNA will be reduced in the second meiotic division. Now, we will see that in detail how it is occurring. Okay, so first we are going to see meiosis 1 and meiosis 2, which all stages are there. Then we will enter into the different stages of meiosis 1, different stages of meiosis 2 in detail. First, we will just see which all stages are there, because it is not difficult for you to remember, because we have already studied in mitosis the same stages it is there in meiosis also. Okay, meiosis 1, in which we can see prophase 1 is there, metaphase 1 is there, then anaphase 1 is there, and telophase 1 is there. So the same thing only we have studied in the case of mitosis, but there is no prophase 1, metaphase 1, anaphase 1, telophase 1 like that. It is just a prophase, metaphase, anaphase, telophase like that we studied. Now meiosis 1, meiosis 2, 2 stages are there. Now that is the reason we are writing meiosis 1, 4 stages are there. Meiosis 2, 4 stages are there. Okay, meiosis 2, 4 stages are there. That is again prophase 2, metaphase 2, anaphase 2, and telophase 2. So these are the 4 different stages of this meiosis 2. So meiosis 1, in that the one cell it will become as one cell which has 2n number of chromosome with the 4C content of DNA will become as 2 cells that will be having n number of chromosome with 2C content of DNA. N number of chromosome with 2C content of DNA. Okay. Now, if we see this, the meiosis 2, in that this cell, it will undergo further division into 2, and we can see it will form 2 cells here, and these 2 cells, it will be having n number of chromosome with C content of DNA. This is n number of chromosome with C content of DNA. Okay, so this n, it is representing the haploid condition. Here this also will form the same way, that is n number of chromosome with the C content of DNA, n number of chromosome with the C content of DNA. So this is normal condition, and it is reduced, this all cells 2n, it became as n is nothing but the haploid condition. Okay, that is the meiotic division. In that we are getting the haploid cells, we are saying. Okay, once again we'll just see this one, then we will enter into different stages of meiosis 1 and meiosis 2. See, the cell which is undergoing meiosis or meiosis mother cell, that is called as meiocyte. Meiocyte is always diploid chromosome containing cell, and when it is undergoing meiosis, it will form 4 cells, and each cell will be having haploid set of chromosome, because our aim in the meiotic division is to reduce the number of chromosome to half, right? So in interphase, anyway, it will pass through G1S and G2 phase. That time in synthetic phase, after that, if we are observing the nucleus, we can see 2n number of chromosome with the 4C content of DNA will be there. How this 2n number of chromosome with the 4C content of DNA comes, that I explained already in interphase of mitosis. And meiosis, it occurs in 2 stages, meiosis 1 and meiosis 2. And meiosis 1 is called as the reduction stage or reduction phase. Meiosis 2, it is equal to equational division or mitotic division. So reduction stage, in which meiosis 1, in which the haploid diploid number of chromosome will be reduced to haploid number. Okay, that is reduction. we will see the two cells will be formed from meiocyte that will be having haploid number of chromosome with the 2C content of DNA, and meiosis 2 in which equational division is occurring. Each one cell it will become as two cells. So two cells it will become as 4 cells, and it will form n number, it will be having n number of chromosome with C content of DNA. C content of DNA is normal for n number of chromosome or haploid number of chromosome. So here you see meiosis, it is divided into meiosis 1 and meiosis 2. Meiosis 1 again it has 4 different stages: prophase 1, metaphase 1, anaphase 1, telophase 1. Then prophase 2, metaphase 2, anaphase 2, telophase 2 in meiosis 2 also. And we can see the 2n number of chromosome with the 4C content of DNA containing meiocyte that will be converted into n number of chromosomes with the 2C content of DNA 2 cells, and each this cell it will undergo second meiotic division or meiosis 2, and it will become as n number of chromosome with the C content of DNA. So 4 cells with n number of chromosome and C content of DNA or haploid number of chromosome with the appropriate concentration of DNA will be present, like that we can say. Now we will see meiosis 1, how it is occurring and how long it will take place. Each stage of meiosis 1, prophase 1, different stages, these all we will study in the next step. Okay. First meiosis 1. Meiosis 1, we said it is divided into 4 different stages. First stage I am writing prophase 1. This is the lengthiest stage of meiosis 1. Okay, so prophase 1 it is again divided into 5 different stages, that is leptotene. It is very important, okay, you have to remember this old name: leptotene, zygotene, pachytene, diplotene and diakinesis. Diplotene and diakinesis. Okay, now we have to see first leptotene. Leptotene, how it occurs. So before entering to this one, I'll just show inside the nucleus, I'll just explain these all stages with the 2 chromosomes, okay, 2 homologous chromosome. With that I'm just explaining this one. So I'm just drawing one nucleus here. This is a nucleus which I'm drawing here, and there is 2 DNAs I'm showing, okay, 2 DNAs when I'm showing, 2 pairs of DNA I'm showing, that I'm showing with orange color and green color. So you see I'm just representing the orange color DNA. This is one DNA with its sister chromatid, and another DNA again with the orange color. This is the another chromatin thread with its sister chromatid. You just imagine this is chromosome number 1. See, in a cell, when a normally a cell when it is not undergoing division, we know it will be having homologous chromosome which has received from their mother and father, right? So you just imagine one orange chromosome that was received from father and mother, and you see it has formed the sister chromatid on this when synthetic phase occurred. So chromosome number 1, like that it is there. This is homologous chromosome, we can say. This is chromosome number 1 received from father, this is received from mother. Both are chromosome number 1 only, and this both has undergone multiplication and it has formed its sister chromatid. Understand this much? Now we will see the next chromosome number 2. Chromosome number 2 I'm just drawing with the green color, okay. So this is chromosome number 2. This chromosome number 2 also is there with the sister chromatid, and it is just like when it is present with the sister chromatid, it look like a single chromosome. This one is a single chromosome, this one is a single chromosome, but the content it will be doubled when we are weighing it. So this is chromosome number 1, homologous chromosome. This is chromosome number 2, which is a homologous chromosome, okay. So this is leptotene stage in which we can see actually what is happening. I'll just say you, leptotene stage in which the DNA will start undergoing condensation. DNA undergo condensation. We have studied in the prophase of mitotic division. the DNA, it will start undergoing condensation, like that we have studied in the mitotic division prophase 1, right? The same way here also, the leptotin stage in which DNA will start undergoing condensation. But the thing is, we are not able to see this nuclear envelope is disappearing, or the centrosome, it is moving opposite pole. These all things we are not seeing. Okay, we are just seeing in the leptotin stage, that is the beginning of prophase 1, and many process it is occurring in prophase 1, in that leptotin stage in which it is just DNA undergo condensation. That is the thing which we are seeing. Now the next one, it is zygote stage. Next stage, it is zygote. I'm just writing here, otherwise we'll not be able to complete in this page. Okay, so I'm just writing here, zygote stage, where the homologous chromosome, it will align parallelly in such a way the bivalent tetrad formation occurs. I'll just show you how it is occurring. Okay, bivalent tetrad formation, how it is occurring, I'll show you. See, again, I'm just taking the orange color for representing the chromosome number 1. See, this is chromosome number 1 now. I'm just going to show the chromosome number 1 there itself, and we can see this chromosome, it will go and align parallelly to this chromosome. It will go and arrange parallelly to this chromosome. You see, chromosome number 1 and 1, it has arranged parallelly. Now you see that chromosome number 2 also will be arranged parallelly, like that. So here we can see chromosome number 2, it is coming and arranging parallelly. This is called as the bivalent tetrad formation. So zygote stage in which we can see the homologous chromosomes arranged parallelly. What is homologous chromosome? Chromosome which was received from father and mother, that is homologous chromosome. Here that homologous chromosome is there with the sister chromatid. Homologous chromosome arranged parallelly, and it form bivalent tetrad structure. Correct. Now you see, bivalent means 2 homologous chromosome. Tetrad, 1 2 3 4, 1 2 3 4, like that tetrad structure will be formed in the zygote stage. Okay, so homologous chromosomes, it will arrange parallelly and it will form bivalent tetrad form. Now you see the next stage, that is Paquetine stage. So here in the third stage in meiosis 1, prophase 1 is the lengthiest stage. Prophase 1 is the lengthiest stage in meiosis 1. Okay, meiosis 1, it is divided into 4 stages. Now prophase, prophase 1, metaphase 1, anaphase 1, telophase 1. In that prophase 1 is lengthiest phase. It has 5 different stages. No other stage has different phases again. Okay, only this one has 5 different stages. In this 5 different stages, Paquetine is the lengthiest. So Paquetine stage in which actually the major process it occurs, that is recombination. Recombination occurs in between the non-sister chromatids of homologous chromosome, occurs in Paquetine stage. Paquetine stage is the lengthiest stage in prophase 1. Lengthiest stage in prophase 1, and we can see the Paquetine stage, the lengthiest stage in prophase 1, where recombinase enzyme will be expressed. Recombinase enzyme expressed in this stage. Okay, actually what is the function of recombinase? You know, recombinase help in recombination between the non-sister chromatids of homologous chromosome. So I'm just drawing that one here. How the recombination it occurs, we will see. They will first form the recombination nodule and all. Okay, so how it will be forming the recombination nodule, I'll show you. See, this is a chromosome number 1 and this is a chromosome number 1. So here the non-sister chromatids of homologous chromosome means what? These two are non-sisters. I'm just representing that one with the red color. See, this one, this one. So these two, I'm just marking these two are non-sisters. So this non-sister, this is non-sisters. Okay, this non-sister chromatids, it will undergo recombination by making the recombination nodule in the presence of recombinase enzyme. How they are producing the recombination nodule, I'll tell you. See, again I'm drawing this one, and the recombination nodule formation, how it occurs, I'll show you. See, again I'm drawing this. Okay, here the next one I'm drawing. Okay, you see the recombination nodule formation, it occurs like this. Okay, it occurs like this. You see the recombination nodule from here to here, from here to here, it has occurred, right? So that way the recombination nodule, when it is occurring, that will undergo recombination. You see, this is the recombination nodule formation occurs in between the non-sister chromatids of homologous chromosome. That is because of what I say, that is because of the recombinase enzyme. Now the next one I'll show you. This is the another chromatin thread or the second chromosome. This also will produce the recombination nodule. How it produces the recombination nodule, I'll tell you. See here, I'm just rubbing this area to show the recombination nodule. Okay, when it is there, it will be just making like this. When it is there, it will be just making like this. Okay, so you see here there is a bulging, it is formed. That is because of the recombinase enzyme. The same way it can occur here also. It can occur anywhere. Okay, anywhere in the in between the non-sister chromatids of homologous chromosome, the recombination nodule formation occurs. So recombinase enzyme help in formation of recombination nodules in between non-sister chromatids of homologous chromosome. Okay, so this is the lengthiest phase, and here we can see their recombination, it occurs. Okay, we'll be able to see the recombination nodule. Here I have just shown the non-sister chromatid, what it is, to show that I have drawn here. Okay, so I did not show the second pair. There is no need of getting confusion because I have not drawn the second pair here. This is just to represent the non-sister chromatids. I have just drawn here. Non-sister chromatid means these two are the sisters, these two are the sisters, these two are the non-sisters, these two are the non-sisters, these two are the non-sisters. Okay, so the non-sister chromatids, to show that only I have drawn here. Now next stage of this one is diplotin. Diplotin stage, in which actually what is happening, you know, this paired homologous chromosome, it will start segregating each other. Okay, so the dissolution of the this structure, it is called as synaptonemal complex. We can say Packetin stage, where we can see a synaptonemal complex formation. Actually not Packetin stage, in zygotin stage itself we will say it as synaptonemal complex. Okay, this is called as synaptonemal complex. Synaptonemal complex, in that, okay, I'm sorry I forgot to say that synaptonemal complex is nothing but bivalent tetrad structure. Synaptonemal complex, in that the recombination occurs with the help of recombinase enzyme. Now next one is diplotin. Diplotin stage, in which the homologous chromosome will try to get apart, or we can say the dissolution of the synaptonemal complex, it occurs. Okay, so dissolution of the synaptonemal complex. Dissolution of synaptonemal complex. What is this dissolution of synaptonemal complex? It is separation of the homologous chromosome. Okay, that is separation of homologous chromosome. when they are trying to separate, what will happen, you know, it is not possible for them to completely separate because it has produced some recombination nodule or recombination has occurred now. There it will not undergo separation. Okay, so there it will form an X-shaped structure where the recombination occurred, where the recombination occurred. There alone it will be joining together, remaining portion it will be separated. Okay, where the recombination occurred, there an X-shaped structure will be formed. That is nothing but chiasmata. That is nothing but chiasmata. Okay, so if we are drawing that one, we can just show like this. This first homologous chromosome, I'm just showing only one place they are joining and staying. Okay, this one got separated like this. So you see here there is an X shape. This is that chiasmata. Okay, one only I am showing, that both one in green color also we can see, or the chromosome number 2 also we can see the same thing. That is nothing but diplotin stage. Now the final stage of meiosis, prophase 1 is diakinesis. Diakinesis, in which this chiasmata, it will undergo terminalization. They will break there. Okay, when they are breaking there, what will happen, you know, I'll just show you. Next one is diakinesis. I'll just write it, after that I'll show you. Okay, diakinesis, where terminalization of chiasmata occur. That means, see here, I just wanted to show that with other color. Green color I'll take. Okay, so green color and yellow color, both yellow color, that yellow color itself I'll take, because we have shown the example this one with the yellow color only, you know. So I'm just showing, see, this will be separated. It will be separated in such a way, this tail is there, no, that has come from here, and this tail is there, no, that belongs to this one, because an exchange it has occurred here. So this is recombined portion, this is recombined portion, this is recombined portion. Because of that, you just imagine in a person, the gamete formation when it is occurring in a testis, the sperm it is forming, you just imagine his father's chromosome, his mother's chromosome. That father's chromosome, it undergone replication, mother's chromosome undergone replication and produces the sister chromatid. So these two are the known sisters. That is the homologous chromosome which has received from father and mother only has that only is the known sister chromatid, right? So you see his father's a part of the chromosome has joined with this chromosome. His mother's a part of the chromosome has joined with this one. Means a genetic exchange has occurred in between the chromosomes of his mother and father in this person's body, right? That lead to variation. This recombination, it lead to variation. Okay, during meiotic division, during meiosis, the recombination occurs, lead to variation in sexual reproduction. You see, sexual reproduction, in that the progeny, it is not exactly looking like his father or mother. That is because of this recombination in between the father's and mother's chromosome in his body, it occurs, that lead to variation. Okay, so that process, it is occurring in the Packetine stage. So which stage in meiosis 1, prophase 1 is responsible for the variation in sexual reproduction? That is Packetine stage. Once again, we are just revising lengthiest one. Meiosis 1, that is the lengthiest stage of this meiosis, and meiosis 1, in this it is divided into prophase 1, metaphase 1, anaphase 1, telophase 1 we said. Prophase 1, again it is the lengthiest stage in these 4 phases. Prophase 1, it is divided into 5 different stages as leptotin, zygotin, pachytin, diplotin, diakinesis. Leptotin stage, actually DNA will undergo condensation, and zygotin stage, where we can see the homologous chromosomes, it arranged parallelly. You see chromosome number 1 and 1, 2 and 2, like that. It is arranged parallelly and it form a bivalent tetrad structure. Bivalent means 2 homologous chromosomes. Tetrad means 4 sister chromatids are arranged. Means two sisters and 2 sisters together it is arranged. That is tetrad structure, and it is called as synaptonemal complex. Next stage is the lengthiest stage of prophase 1, that is pachytin. There the recombinase enzyme expression will be there, and recombinase enzyme that will help in making the recombination nodule in between the non-sister chromatids of homologous chromosome. Non-sisters are these two are non-sisters. So the non-sister chromatin thread which is coming close each other, that will make the recombination nodule with the help of recombinase enzyme. Okay, then after that it will undergo diplotin. It will enter into diplotin stage. Actually this recombination, as it is occurring in this pachytinate stage only, variation it occurs during the sexual reproduction. Now next one is diplotin stage, where we can see homologous chromosome, they will try to get apart, but the thing is where they have joined during recombination, there it will be just joined together and staying as an X-shaped structure, that is called as chiasmata. So chiasmata formation occurs in the diplotin stage, and diakinesis in that where chiasmata has formed, it will just break that area and exchange the genetic material in between the non-sister chromatids of homologous chromosome. What they will do? Exchange the genetic material in between the non-sister chromatids of homologous chromosome they will do, and they will just terminalize the chiasmata. So this diakinesis, it is the end of the prophase 1, and end of the prophase 1 where we can see the chromosomes that will get completely condensation, meiotic spindle and all, it will be assembled to prepare the homologous chromosome separation or chromosome separation. You just understand. By the end of the diakinesis, when we are looking, we can see nucleolus will not be present, nuclear envelope will be broken down. Diakinesis, that is the representation of the starting of metaphase, or it is a transition state to metaphase, like that we can say. So diplotin stage, in which the remaining features of prophase, late prophase, like nuclear envelope disappear, nucleolus will disappear, and we can see the cell organelles, it will start disappearing, complete condensation of DNA it occurs, mitotic spindle it will be arranged. These all occurs in the diplotin stage at the late prophase. Okay, so this is all about the prophase. Now you see, we'll just revise quickly from the first page. See, we started with meiosis. Meiosis is nothing but reduction division. Meiosis, it occurs in a diploid cell, that is for reducing the number of chromosome to half. This is for maintaining the uniformity in chromosome number in generation after generation. And we can see the meiosis, it occurs with the diploid cell, and zygotic meiosis is there, sporic meiosis is there, and gametic meiosis is there. Zygotic meiosis occurs in thallophyta. Sporic meiosis, it is seen in bryophyte, pteridophyte, gymnosperm and angiosperm. Gametic meiosis, it is seen in animals. Now next one, meiosis, which cell is undergoing meiosis, that is called as myocyte. If the zygote is undergoing meiosis, zygote is myocyte. So thallophyta, in that zygote is myocyte. Myocyte, it is diploid, undergo meiosis to produce 4 haploid cells. Now we can see whatever this division it occurs, it will pass through the interphase, and interphase in that G1s and G2 phase it will be following, and 2n number of chromosome with the 4c content of DNA will be present at the last stage of the interphase. Meiosis it occurs in 2 stages as meiosis 1 and meiosis 2. Meiosis 1 is a reduction stage. 2 cells will be formed with haploid set of chromosomes and diploid content of DNA or 2C content of DNA. Meiosis 2 equational division occurs. Each cell from that two cells formed in meiosis 1 will undergo meiosis 2, and it will become up as two cells, means total 4 cells with a haploid set of chromosome and haploid content of DNA. Now, meiosis 1, it has 4 different stages. Meiosis 2 has 4 different stages. Prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 2, metaphase 2, anaphase 2, telophase 2. Prophase 1, meiosis 1, in that the diploid cell with 4C content of DNA will be divided into 2 cells or haploid number of chromosome with the 2C content of DNA. And meiosis 2, in that these 2 cells which is formed as haploid cells with the 2C content of DNA will undergo second meiotic division, that is equational division, and it will be just equally dividing the content of DNA into 2 cells, and it will become as n number of chromosome with the C content of DNA. Like that 4 cells will be formed. Now we are studying the prophase 1 of meiosis 1. So prophase 1 of meiosis 1, it is divided into 4 different stages, I said, right? Prophase 1 only is the lengthiest stage. Okay, prophase 1, remaining everything it is just like mitosis, don't worry. Only this portion it is little big, but it is very important for us. Prophase 1, it is divided into 5 different stages: leptotene, zygotene, pachytene, diplotin, diakinesis, like that. It is divided into 5 different stages. So in that leptotene stage, in this, just a condensation of the DNA it occurs. So there we can see chromosome number 1 and 2 I have just shown. 1 it is with orange color, and 2 it I have shown with green color. And next it enter into zygotene stage. Homologous chromosomes, it will be arranged parallelly and it form a bivalent tetrad structure. That is nothing but synaptonemal complex formation. It occurs in this one. Now next one is pachytene stage, where that is the lengthiest stage in prophase 1. That will be having an expression of a recombinase enzyme. That recombinase enzyme will help in formation of recombination nodules in between the non-sister chromatids of homologous chromosome. Recombination nodule formation, it is like this. Okay, and it will undergo recombination. Because of this pachytene stage only, in sexual reproduction variation occurs in the progeny. Progeny varies from father and mother. That is because of this pachytene stage recombination. Next one is diplotin stage, where the dissolution of synaptonemal complex occurs by separating the homologous chromosome. Where the recombination occurs, there one X-shaped structure will be formed. That is called as chiasmata. Okay, chiasmata is a characteristic feature of diplotin stage. Many cells which is like oocyte of many animals, or like vertebrates, they will get arrest in diplotin stage. Means after the chiasmata formation, no, they will be just getting arrested in diplotin stage. We can see that in the case of human beings. We can see in the case of amphibians, oocyte, we can see. We have already studied about Lamb-Brush chromosome. Lamb-Brush chromosome, that is the chromosome which is getting arrested at the diplotin stage of prophase 1 of meiosis 1 in the amphibians oocyte. Like that we have studied when we discuss MCQ in the material. I told about this Lamb-Brush chromosome. Okay, that is arrested in the diplotin stage. Even human egg is also getting arrested in the diplotin stage. The primary follicle in that oocyte, it is getting arrested in the diplotin stage. That you will study in 12th standard. Okay, now next one is diakinesis. That is terminalization of the chiasmata. Terminalization of the chiasmata, in that X-shaped structure where it is formed, there they will just break that that region, and genetic exchange, it will be occurred in between the non-sister chromatids of the homologous chromosome. Okay, so this diakinesis stage, the terminalization when it is occurring, by the time the prophase 1 will be completed. Prophase 1, when it is ending, we can see the nuclear membrane will be undergoing disintegration, and the nucleolus will be disappeared, and we can see the cell organelles and all, it will be disappearing. Chromosome, it will be completely condensed, and we can see see the spindle apparatus and all, it will be formed. Okay, that you just write according to the note which I have given in prophase of mitosis. Okay, and you just listen and you can write this also, there is no issue. And that is the end of the prophase 1. And we can see by the diakinesis it is showing the entry to the metaphase 1. Next stage, it is metaphase 1, that we'll study in the next class.